Last week, we talked a little bit about bacteria, the Monera kingdom, and we talked about some bad bacteria and some good bacteria. There was one that I left off my list of good bacteria because I wanted to go into more depth about it, and that's what we're going to talk about today. But before I can tell you about that bacteria, I need to tell you what a transgenic organism is. Transgenic means that you have transferred or transplanted some gene out of one species into another species where that gene usually doesn't occur. The first one that I ever saw was a tobacco plant. The scientists have taken the gene that makes a firefly's butt glow, right? You know what I'm talking about, lightning bugs. These little fireflies, they have a glowing rear end that they flash at each other in the middle of the night, and that's how they communicate. And there's a gene for that, right? A little segment of their DNA that makes their butt glow. So scientists had taken that glow butt gene out of the firefly and put it into tobacco plants. And it was very eerie. I walked out into an entire field full of those tobacco plants, and they were all glowing green in the middle of the night. That was a long time ago, and we've done a lot more since then. So what is a gene? Well, a gene is a little segment of DNA that codes for some trait. You have a gene for your eye color. You've got a gene for your skin color, a gene for your hair color. There's a gene that tells you how tall to be, and a gene that tells you whether to have freckles or not, right? So all these little genes control all these traits that you have, and those genes could be chemically cut out of there, like you were taking a pair of scissors and snipping it out, and then move it into another species where it doesn't usually occur. And they've done a lot more since the glow-in-the-dark tobacco, even with the glowing gene. They've taken that glow gene, and here's a little cartoon representation of it. You cut that little gene out of the strand of DNA, and you insert it into a mouse embryo, and then as that mouse grows up, their skin fluoresces, just like the firefly's butt did. And they have made glow-in-the-dark mice that are green, yellow, red, and you can't really tell that well until you turn the lights off, but then they glow in the dark just like a firefly would. One of the first things that I ever saw this technology used for commercially was the neon fish that are for sale at the pet store. There were scientists, I think they were using the neon color to mark certain fish in a population that they wanted to watch so they would stand out. But they realized pretty quickly that they were just pretty as well, and people would pay for them. So they started selling them to pet stores. That's a transgenic organism. Now, you may think, well, that's weird, and I don't know if I'd want one of those things because it's kind of, Frankensteinish. I don't know. But in reality, you eat genetically modified stuff almost every day. Almost every bit of food that you buy in the grocery store, you eat at a fast food restaurant, has been genetically modified and has genes transferred into it from other organisms. Corn, for instance, has a gene transferred into it from bacteria. And that bacteria is a toxin to certain little caterpillars that like to eat corn. Now, you could spray that bacteria all over your corn, but then you got bacteria all over your corn. That's probably not good for anything. So what they've done is they've taken that little gene out of the bacteria that kills the caterpillars, and they transfer it into the corn itself. It doesn't hurt us, but if the caterpillars try to eat it while that corn's growing, they die. So, and they've done this to all kinds of food that you've been eating. You can take a gene that causes muscle growth, and instead of just having one gene for it, you can insert multiple genes for muscle growth and have huge muscle-bound cattle that have extra muscle genes inserted into them. Imagine if the bodybuilding world finds out about that, right? One of the weirdest ones I've ever seen was someone had transferred the gene for making spider webs into the goat's milk production gene, so that when you milk these goats, you not only get milk out, but spider web comes out as well. And they're using that to make like silk clothing that's usually really, really expensive, but you can get a lot more of the silk out of a goat than you can a spider because they're so much bigger. Now, you can also do some really weird stuff like take entire genomes, not just a gene, but like half of a lion and half of a tiger's genome and smash them together in one egg cell and create a liger that's half lion, half tiger, or a geep that's half goat and half sheep, right? So, and there's a lot of potential uses for this that we haven't even tapped into it yet. I mean, imagine inserting a gene for growing an ear into a mouse's skin cell and being able to grow an ear to replace somebody's ear that got cut off, right? So all this stuff's just really interesting. But wait, what does this have to do with bacteria, right? Well, let me get to that. So most of us could go to the grocery store and just load up on candy and cake and sweets, and we could eat until we got a stomach ache if we wanted to, but we'd be fine a few hours later. That is not true of somebody that has diabetes. If they eat too much sugar or their blood sugar gets out of whack, it's life-threatening. And it is that way because their body is not making a certain chemical that usually stays in our bloodstream and helps regulate sugar in our bloodstream. It's called insulin. Your pancreas makes it. Their body doesn't make enough of that. And so their blood sugar levels can get off balance, and it can become a life-threatening situation. Now, historically, back in the 1920s, 30s, we were getting our insulin from pigs because pigs have insulin in their bodies as well. So you'd take a bunch of pig farts, grind them up, and make kind of a soup out of it, and then try to isolate the insulin out of those pigs. But that required you to kill the pigs and catch the pigs and feed the pigs, and that's expensive, right? And a lot of time to do that. Well, eventually some scientists got the bright idea that they might be able to make a bacteria produce that insulin. And what the scientists found out was bacteria not only have a large strand of DNA that has most of their chromosomes in it, but often they also have a separate, smaller, little circular strand of DNA called a plasmid. And sometimes this will code for some special ability that that bacteria has. And they sometimes trade it between each other. So those little bacterial plasmids are not absolutely necessary to be coded in a certain way. You could take it out and the bacteria would probably still survive, or you could replace it with something. So the scientists got the bright idea that they could put a human gene into that plasmid. So what they did is they figured out a chemical way to snip, almost like a pair of scissors, to snip that DNA strand and then jam a human gene into the little ends that they just cut apart. Now, it is a complex process, and if I was smart enough to figure out how to do that chemically, I wouldn't be working as a teacher. I'd have a good salary. But you can just think of it like snipping it with a pair of chemical scissors, right? You take the little bacterial circular plasmid, you cut it with little restriction enzymes. Those are the scissors. And then you take the human insulin gene and you stick it in, and it will latch on to those free ends of the circular plasmid. And it kind of glues itself back together with some other little enzymes. So, and this is an actual electron scanning microscope picture of it. You can see the bacterial plasmid that's been cut, little human gene right there in red, and then you can see it put back together. So those little circle things are called plasmids. And that's what you're going to be making me today. You're going to be drawing a map of a plasmid. There's more instructions in the lesson itself. But when scientists have made something like that, they've made their little plasmid, they've inserted a human gene, and usually a couple of other genes that they had to stick in there too. They want to make a map of that so they know where they put stuff in that bacteria. And they might insert 3 or even more genes in that little plasmid, and they make what's called a plasmid map out of it. Just a circle, and that circle shows where they stuck those genes. Now, this one's just showing you can stick 1, 2, and 3 genes in, but a real plasmid map looks something like this, right? Now, I know that looks complicated, but don't panic. You're making me one of these today, but it's not going to be anywhere near this complex. If you pay attention to what's going on in this one, notice there are 3 arrows, a red one, a green one, and a purple one. Those arrows are telling you the important stuff. Like these things here, I really don't even know what those are. But I do know what the arrows stand for. I figured it out. It's pretty technical language on this real one, but yours won't be that complex. So what those arrows are showing you, the green one is showing you here green fluorescent protein gene. That's the gene that they've stuck in this bacteria that'll make it glow in the dark. So they've probably gotten that from the firefly, and they've stuck that gene here in this bacteria to make a glow in the dark bacteria. They've also stuck one in called ampicillin resistance, and I know what that is. That is something to make them resistant to the antibiotic cleaner that the scientists are going to use to clean their lab. You got to clean up if you're going to grow bacteria in your lab. You don't want it to get out of control, but you also don't want your cleaner to kill the bacteria that you're working on because they're very valuable. So they've made this one resistant to the antibiotic cleaner that they're using in their lab. And then this final one here, this arabinose operon, that is a gene that they put in there to make it eat a certain type of sugar, probably because that sugar is cheap for them to feed it, and they want it to be able to digest that certain sugar. So they're making a bacteria that is resistant to their cleaner, can eat the cheap sugar that they're feeding it, and it'll glow in the dark. It's an interesting little bacteria, but it's not doing much, really. There are some other ones, though, that they've designed, kind of like the insulin bacteria. That insulin bacteria, once you get it made, will produce insulin all day, every day. And you can easily get it out of that bacteria and then start a new batch growing. It was way cheaper than trying to grow those pigs and get the insulin out of them. But they have used bacteria for other things as well. For instance, they've designed some bacteria that will eat oil. And what they can do with these is go out where there's a big oil spill on the ocean and the oil is threatening to kill the sea life and the birds that use that ocean. And they can spray these bacteria on the oil slick, and those bacteria will sit and eat that oil and turn it into something much less harmful to the environment. They also have some, and I heard about this from a military guy who said they had a tanker truck that they could pull in somewhere where it was just sand, and they could spray these bacteria on the sand, and within a day or two, that sand was almost like concrete, and they could build on it. So this is, I looked this up later, is bioconcrete. These little bacteria will secrete something around themselves. It's almost like a concrete. And so if you build a sidewalk that contains these little bacteria and you accidentally drop something on it, or it cracks or it weathers, these little things will reseal that concrete. It's like self-healing sidewalks, right? And that gave me an idea for the bacteria that I designed as an example for your lesson today. Now, I don't want you to copy my ideas. There's a million things that you could use, and you don't have to use the two things that I used. But the idea of something glowing in the dark and the idea of concrete building bacteria gave me the idea that I used for my example, and you'll see it in a few minutes. But I want you to use something unique. I don't want you to use my idea. So don't use the oil eating idea or the insulin making idea or the concrete idea, because I've already given you those. But the sky's the limit here. You think of a bacteria that you want to design, come up with something inventive, surprise me. All right? Good luck. We'll talk to you later.